Hello, everyone. I hope y'all are doing well. We're here to now begin our discussion on Protista. So up to this point, you should be very knowledgeable on bacteria, the eubacterians, the archaebacterians, those prokaryotic groups. We're going to progress now in the evolutionary process into a very diversified group that we call Kingdom Protista. So Protista is this jumbled up mess of species. Some people, and I like to consider this also as a junk drawer kingdom. I don't know if y'all have a junk drawer like this at home, sort of random stuff that doesn't really fit anywhere else. It's convenient to maybe find here, that kind of stuff. This kingdom, this junk drawer kingdom, this sort of orphanage kingdom represents the Protistan category, right? Just jumbled up mess of stuff in here. So Kingdom Protista, again, a very diverse assortment of organisms, not a common theme, not a common characteristic that they all share aside from being eukaryotic. That is the only sort of unifying quality that Protistans share. They are eukaryotic celled organisms. So that takes us from the prokaryotic bacteria, now a big jump in the evolutionary process that is the eukaryotic celled protistans, right? So again, jumbled up mess. Some are going to be single celled, one cell, so they're microscopic. Others, like some huge algae, are multicellular, almost 60 meters in length. So again, that's a huge difference from a microscopic single cell to 60-meter large organism. Huge distinction in body size and in body arrangement, complexity for protistans. Some are going to be photosynthetic. They convert sunlight into energy. Others are going to go out and hunt, so they're predatory. So again, we have this mixture of species in the Protistan category. Oh, the phylogeny is kind of rough, right? So I'm not going to get into all the detailed phylogenetic groups of Protistans. I'm going to kind of go rapidly through this. We're going to break it into three categories. So protozoans, what they consider the animal-like protists. So animal-like because they move, they can swim, they're heterotrophic. They go and hunt, they eat other things. They're not photosynthetic per se. So the protozoans, right? The algae, which are very plant-like. Why are they plant-like? Because they are photosynthetic, right? So they convert sunlight into to carbohydrates, releasing oxygen. So again, these subdivisions, they're not an official taxonomic group, but they're gonna kind of give you a hint as to how these protists behave, right? How do the protozoans feed? How do the algae feed? And then the slime molds, these are fungus-like. So these are gonna be saprobic. They're gonna have to be in direct contact so they can absorb nutrition, right? So the fungus-like, the plant-like, and the animal-like protists, right? So the protozoans, the algae, and the slime molds. Again, not a true phylogenetic categorization. We're looking at somewhat morphology, kind of, sort of, but more of nutritional-based subdivisions that we're looking at here. How do they acquire nutrition? So protozoans hunt, algae photosynthesize, slime molds go and they are in direct contact and they're absorptive feeders, saprobic feeders. So for protozoans, we look a little bit at their morphology, their shape. And we established, if I backtrack, they're able to move. They're very mobile. So, well, if you're very mobile, what do you utilize in order to move? And that's going to be the distinction for these morphological groups. We call the flagellates. The flagellate would have a large dominant little tail. A ciliate would have a lot of tiny little hairs all over the cell. Sarcodines are going to be sort of this pseudopod ability, or the ability to form pseudopods. And the apicomplexans, I'll show you a picture of those in a bit. So again, what do they have in common? They're all eukaryotic, and they all go out and hunt. They have the ability to move and find prey. So these protozoan categories. So the first one is actually a kind of a curveball, kind of an odd group. It kind of epitomizes then this complexity of the protist. So we established that these are flagellates, and this is what we call euglena, right? Euglena is a very common representative of a flagellate. Some people categorize them more as algae, right? They are possessing chloroplasts. And we know chloroplasts are the organelle that allows for photosynthesis. So this is kind of a strange position to put euglena in the protozoan, the animal-like groups. They have basically like an eye, a very primitive eye spot, but they photosynthesize. So where does it go, right? We know it's not a bacteria. We know it's not an actual animal. We know it's not an actual plant. So it gets dumped in this orphanage, this junk drawer kingdom that we call Protista. Right? A couple of relevant flagellates, and I'm going to take a moment to discuss these because we have a few researchers here at UTEP that work, they've spent their whole careers studying two species, right? Giardia lambia, I should say two groups, right? Giardia lambia and several species of Trypanosoma. So I kind of hyped up the Bridges research program. Maybe if you work at UTEP, you might end up working in some of these labs, right? Dr. Sid Doss working with Giardia lambia, and we have a couple of researchers that are working with Trypanosoma, and I'll elaborate on them in a bit here. So pathogenic. So we know that they're pathogenic. They're going to cause some sort of disease. Right? So Giardia is it's a protist, not a bacteria. It's a protist that if it gets in the system causes intestinal distress. You're out hiking, you drink what you thought was clear crystal water, and basically you got sick, right? So you have diarrhea, you have cramping. This is the pathogenicity of Giardia. Trypanosoma, there's multiple species in the New World and the Old World. And in Africa, they're responsible for African sleeping sickness. African sleeping sickness is a condition that basically these individuals lose consciousness, they go in a comatose state. So in the past, people thought, well, they're sleeping, right? So they're not really sleeping. It's a African unconscious sickness, something like that, right? So it's not a nice condition. In relevance to Trypanosoma and relevance to African sleeping sickness, we go back to some ecology. So we know that insects, particularly flying insects, parasitic insects, are very good vectors. We learned vectors before in bacteria, now we bring vectors back. So the tsetse fly is this fly here that basically is a blood-sucking insect. It bites an infected host and it transmits then that infection to other hosts. Right? So again, the actual causative agent for the sleeping sickness is Trypanosoma. The vector that's transmitting Trypanosoma from different hosts would be the tsetse fly. So closer to home, right? Not in Africa, but closer to home, we have different species of Trypanosoma, and we have a different species of vector, right? Here we have, in the United States, we have these reduvid beetles. You might know them as kissing bugs. Kissing bugs or reduvid beetles are known vectors of Trypanosoma. So if you were to be unfortunate enough, right, you're in an area where there's chagas, or I should say kissing bugs, that are transmitting Trypanosoma. And if you get infected with Trypanosoma, you could develop what we call Chagas disease. That's in Central America, South America, and now into North America. So kissing bugs, why do they call them kissing bugs? Because they're out parasitizing at night, and they would basically search for thin skin in which they could puncture and draw blood. So thin skin of the eyelids, thin skin of the lips, right? So you're sleeping there, mind your own business. The kissing bug arrives, gives you a little kiss, steals some blood, but could also introduce into your system now these Trypanosoma parasites. And if you are infected over time, you could develop then Chagas disease. So a lot of research is being done with the actual parasite, the Trypanosoma, and the corresponding disease, Chagas disease. So we have two renowned faculty at UTEP, Dr. Igor Almeida and Dr. Rosa Maldonado, no relation to me, but they both have sort of committed their whole careers, right? They have a passion for learning more about curing Trypanosoma, understanding its life cycles, understanding the physiological effects it has on the body. And these are some of the conditions that would occur if you're infected with Trypanosoma. So we see a very sort of enlargement of the ventricle. We see a very big weakening of the myocardium, of the heart muscle. So the heart basically becomes an inefficient pump. We see a tremendous amount of swelling and inflammation on the digestive tract. Right, so both of these would be very, very detrimental to your health, right? And you're looking now at cardiovascular issues and digestive issues, and these are coming on simultaneously. So these are basically conditions that could lead to death in a very chronic, long-term type of situation. So if you do get the chance to go to UTEP and do some volunteering or some actual research, potentially, communicate with Dr. Almeida or Dr. Maldonado and let them know that you're interested in this kind of stuff, and you would be in very good hands with both of their labs there. Right? So pathogenic flagellates, and we could go on and on and on covering different species, but we're just trying to introduce this category here. So there's again the actual causative parasites, these trypanosomes that will swim around in the blood and blood plasma. And we shift gears now. We look at a second category of protozoans that we call the ciliates. So ciliates would have all of these little hairs, all of these little cilia that allow for a very, very refined movement. Maybe not as fast as a flagellate, but much better control. We can go forward and backwards and sideways, and a lot more control to their movement here. So the representative for the ciliates is going to be this very complex cell we call Paramecium. Paramecium is complex because of some genetic aspects, right? We have in this group a large macronucleus and some smaller micronuclei. And the genetics behind this is kind of interesting. Why would we need multiple nuclei? Right? How would that help the cell? Is it for backup? Is it for storage? Are we transcribing and translating certain types of DNA and not other types of DNA? So again, a lot of interesting genetic aspects with these very complex single cells that we call Paramecium, right? The representative of our ciliate group. So you can see all the cilia. Let's see, so cilia is going to be also not just in one dimension on the outside, it's three-dimensionally around the cell. Here you see in like that lime green, the macronucleus, and in yellow we see the little spots, which would be basically the micronuclei. And so complicated little cell. Our sarcodines are going to be represented by a very common, very famous type of protist that you might know of. We call this Amoeba, Amoeba proteus. So Amoeba proteus is a very flexible little protist. If there was a gymnast or a yoga type of protist, it would be Amoeba. Right? Amoeba is so flexible. Its membrane is very flexible. It can kind of extend and protrude in all different directions. So all of these are the same species. They just look different because they're so flexible. Their cytoplasm can be streamed into different positions here. So this flexible cytoplasm, or I should say this flexible membrane, very liquidy cytoplasm, enable the amoeba to form pseudopods. Pseudopods are these little like arm-like extensions. Pseudo means false, pod means foot. So like a false foot, a little false extension. And as we stream the cytoplasm, we stream it, and it starts to form like a little arm this way, and maybe another little arm that way. And they kind of extend until they can go around and wrap. So kind of like a series of photos that are showing the elongation of the pseudopod here. So pseudopods are getting closer. Eventually they're gonna connect and bring in this prey item into the cell. So kind of the action sequence: extending the pseudopod, continual extending, developing another one, and eventually, again, that's how they feed. This is not necessarily a rapid process, but it does enable the amoebas to kind of acquire nutrition that way. All right, so we have established our flagellates, one long dominant locomotory structure. Then we have the ciliates with all these little hairs, and then the very flexible pseudopod forming sarcodines. So now we're gonna kind of look at a very complex type of protists, and it gives us a hint in the name. They're complex. They're called the apicomplexans. So apicomplexans are gonna be obligate parasites. So they have to survive as parasites. They don't have an option. If they're not being parasitic, they cannot survive. So it's a complex type of situation. And then they're not going to just parasitize one host. They have to parasitize two in order to complete their life cycle. So they're obligated, they're forced to parasitize two very distinctive species in order to complete their life cycle. So that puts kind of a hardship on their life strategy, and it makes them become very, very adaptable and very, very sneaky in which they can parasitize different hosts. So a representative here is the one we call Plasmodium. So Plasmodium is the representative of our apicomplexan here. So Plasmodium is of medical significance because it causes malaria in humans, right? So it does cause this disease, malaria, which is not a nice condition, and it does kill many individuals on a yearly basis, right? So something not to be taken lightly. So let's start here. So this person's minding their own business. They're at the picnic. They're out at, just out about. And we have then this infected mosquito, right? A very particular type of mosquito that we call an Anopheles mosquito. So the Anopheles mosquito is a very, again, a particular type of mosquito that can be the vector for Plasmodium. So the infected mosquito bites, steals blood, but introduces now Plasmodium into the human system. So Plasmodium is then picked up by the liver, which is what's supposed to happen. The liver is supposed to detoxify foreign sort of aspects. Within the liver, we have then some damage that happens. So the liver is unable to destroy Plasmodium. But if you notice, Plasmodium comes in looking this way, it changes, it modifies, and now it causes lysis of the liver cells. So now the human liver cells are rupturing, which is not good, right? You have a destruction of liver tissue and again, a change in strategy. So our immune system is starting to look for this, and now they changed into this form here. So they don't stay vulnerable to the immune system. They quickly find a red blood cell and enter into the red blood cells. So when they continue their change, their maturation process, and then they burst out of the red blood cells. So again, you can see two very harmful situations: the rupturing of liver cells, and now the rupturing of red blood cells. Both of those are medically significant consequences here. So red blood cells are rupturing, and that's very convenient because there's other mosquitoes in the area. and they're still feeding, they're still parasitizing. So when this mosquito bites and sucks up blood, it can suck up this new form, this gametocyte form of the parasite, the Plasmodium. Now, once they're in a mosquito, now sexual reproduction happens in the mosquito. So in essence, Plasmodium is using the human as a nutritional source, but they're using the mosquito as a reproductive source. Right? So once they reproduce, they start the next generation, and now the babies are sort of sent into the human again in this cycle. Right? So again, a pretty elaborate cycle, difficult to treat. Right? Wherever we have humans and mosquitoes, there's always a potential risk. Right? So Anopheles is a very prevalent mosquito. Humans are a very prevalent species, almost 8 billion on the planet. So I don't really see a quick solution to this problem. I suspect malaria will be around for a long time. There's a lot of poverty. There's a lot of areas where humans are just constantly being bitten by mosquitoes. So again, not a condition that we're going to see go away in any time soon. Now there is medication. There's prophylactic medication, quinine, things like that, that can be used to treat or to prevent to prevent the transmission or the acquirement of Plasmodium. So be aware, if you travel, if you're in the military, that kind of stuff, you will be eligible for some of these prophylactics too. So again, this is what normal human blood should look like. There's our red blood cells. There's our white blood cells. And here we have a different situation now, red blood cells that are infected with Plasmodium. So Plasmodium lives inside of the red blood cells. And to contrast, Trypanosoma, the Chagas disease one that we covered earlier, that one lives outside the red blood cells in the plasma. So different strategy. So Plasmodium would live inside the red blood cells. Trypanosoma would live in the white area outside the red blood cells. So again, they are diagnosable if you know what you're looking for, if these tests are done to treat or to identify the presence of these parasites. So with that, let me stop and I'm going to upload this one, and I'll finish off Protista with the next discussion on the algae here, right? So let me get this one uploaded.